Okay, why don't we get started? A couple of announcements before we get to this, and one of them you'll be quite pleased with. I'm moving the exam to October 2nd, 1 week delay. It's not because of you, it's because of my schedule. So I won't be able to give it on this next Tuesday. So if you prepared, you're in great shape. If you haven't prepared, I just saved your butt. So, if you have not caught up, this is the weekend to do it. And also, those problems that I posted, the assessments, remember, the first assessment one is out of here, it's a bad post, so you have to completely ignore that one and do 1X instead of 1. So, because I'm going to erase the points that people have. Yes?
Are you going to add more material to the test, or is it still just the...
Yeah, that was the good news. This is the bad news. But no, it's actually just a small segment that, because we're just stopped in the middle of this mess, and so it's really hard. That was a bad place to stop anyway. So we're just going to finish lecture 6 material. But not lecture 7 is not, and I'm going to draw a bright line. It's going to be a PowerPoint picture that says this is the separating. So it's not a lot extra. It's just to help you understand this mess right here. It really didn't. It's not very fully explained. Okay, but it is a little bit more material. It will have a full week of no new material for the exam. Okay. Briefing out. So the other thing about those assignments. The reason we did this sort of practice stuff with you using the Dropbox and all that, although this is the assessment, or more like multiple choice tests, you have to be able to do it before the deadline. At the deadline, it goes down, okay? So if you're smart, what you'll do is you'll try to do it early. Do not wait till the last minute. Because if Blackboard goes down, that's your fault. That's just the way this works. Electronic submissions are... I don't know, they don't always work. So that means you cannot afford to wait to the very last minute to get those done. If you have problems, you need to contact them or one of your group members post to Beyonce, get some help, but do it well before, I think it's what, 11:30 Saturday night? I know you'll all be studying cell biology. Yeah, not really. But, so just do it early, because there will not be, I didn't get it, or I didn't see the assignment, I didn't know it was due. That's not an excuse. And besides, these are optional. You should really do them, but there'll be more points than this to be earned. So if you screw it up, just take that as a notice to do better next time. But you have to have those assignments in by the deadline. Okay. All right. A couple things to the graduate students. Since we're not having the test next week, what I want to do is make sure you get your papers, your free choice of papers into me as early as possible. Particularly, this is true for Nancy and Daniel because you're the first presenters. So if you can get them to me this weekend, that would be wonderful. And I can make my selection. You can go get those because you guys are up first. Okay. All right, so that was just for the graduate section. And so August 2nd, that'll be your exam. The departmental seminar on October 3rd is relevant to cell molecular. The speaker is my guest at the medical school, Pablo Artigas. He works on the sodium pump. And that's the next topic that we're going to talk about, and that's membrane proteins and membrane transport. So I highly recommend that you go to that. I probably will set up some points for attending, and you'll have to write a little brief paper, 1 page or so, on what he said. He's interesting, he knows his stuff, and you'll need to know that sodium pump backwards and forwards anyway, so it's good timing for all of us. So that's October 3rd, 4 to 5 p.m. There's actually refreshments somewhere in 102, 103 before that, so if you can make it, do come. And he's also looking for people, good undergraduates, to work in his lab. That's one of the reasons he wants to come over is recruit. So if you find his work interesting, you should read up a couple of his papers, go to his website at the med school, read them, and then set up an appointment. But don't go in and talk to him if you haven't read them, because you need to know what you're talking about. Okay? But he's a really nice guy. Okay. Anything else? Any questions? I'm sorry? Where's Savannah? You'll go ahead and have it? You'll still have it on Monday?
Yes.
Okay, so that means you have to get ready to have good questions. Is that your question as well?
Keeping the date of the discussion of the
I'm sorry, I couldn't hear.
I just repeated what it was.
We are going to have it on the 24th. So. Earlier the better. Actually, this is the way it should work. But I'm not trying to fool you into studying early. This just happened. So I decided last night. Yes? It's the Artigas lectures. The Department of Civil Education don't know we're open to you. But I try to alert you to those that are cell molecular nature. Some of them are ecology. But it's room 101 and 5. 4 to 5. Yeah. Refreshments at 3:45, something like that. Okay, so. All right, so we have this gobbledygook here, and okay, so this is the fully assembled, stabilized pre-initiation complex. The reason it's stabilized is that there's some transcription factors, activators that are bound to enhancers at various points that are holding on to this thing to make sure it's stable. And as long as this thing can be stable, it's going to transcribe. Okay? But actually, it ends up leaving most of the pre-initiation complex subunits behind. And so once this starts moving, it's going to elongate and start transcribing RNA, messenger RNA in this case, reading the antisense strand. It's also called the template strand. It's also called the coding strand. So you need all those terms are interchangeable. Okay, so We're going to talk about how these gene activator proteins work together, because it's the unique combination of a bunch of similar guys acting at the promoter that cause differential gene transcription. That's the big concept. It's not unique proteins for this gene, unique proteins for this gene. It's a unique combination of similar proteins. Okay, so they have to work in combination. Okay, so let's there's a little question here. This is actually a student question a few years ago. This goes back to how well do you understand the review of the background material for gene transcription. So take a look at this. It's not a multiple choice, so see if you can answer it. Okay. Anybody want to take a try? This one not a bad question yet.
Well, one thing is that the RNA pull to be identified first is the transcription factors B, which binds to the autoboxyl.
That's right. Okay, but there's which strand is it reading? It's kind of a trick question. It doesn't read one strand. It reads the unique combination. Remember those? That's why I showed you those little beads, like abacus things. It reads both strands. It's reading the combination of both strands, and that's also what this is doing. Pole 2 has got to figure out where it is, and it's got to decide which one is the template strand, which one is the non-template strand. But it doesn't bind there because of that. It binds there because it's a duplex, both strands. It recognizes the TATA sequence and its complementary sequence, which would be AT-AT, correct? So it sits down on the railroad tracks, not just one rail. Okay. And then when it decides, it can tell by the orientation of the other transcription factors that this is the coding strand, this is the template strand, this is the antisense strand, and the antisense strand is running 3 to 5. It's going to make the transcript 5 to 3, right? The new transcript coming off is 5 to 3. The first thing that emerges is the 5' end, and what happens to that 5' end as soon as it appears? I said it. It's capped by the 5' cap instruction. Okay? And that's to keep it from being degraded. So that happens actually co-transcriptionally. Okay? And then the actual splicing of the sequence occurs as pole 2 moves down the sequence. And the RNA transcript is as soon as it emerges, the spliceosome grabs onto it and starts to go, okay, this is an intron, this is an exon, this is an intron, so just cut them out and splice them. Okay. Good. Okay, so here's the basic concept about synergy. Okay, if you have one, this would be like one enhancer, one activator. This is a different enhancer, a different activator. So you get X amount of transcription when this guy binds to that one control element. You get 2 units when it binds to this one because this one is a little stronger in promoting transcription than that one. So you can have individual strengths of promotion. But when they both bind, what you get is synergetic increase in transcription. It's much more than the sum of the 2 parts. That's why, another reason why combinatorial control is important. It dramatically amplifies the degree to the strength of promoting transcription. Plus, because it requires lots of different proteins to be bound here, it makes there only one unique combination that's going to work and work the most efficiently. Okay? All right, so what we want to be able to do, we've done this, we talked about how you construct a DNA binding domain, we want to construct a switch, and then we want to talk about, we've talked about some construction, but I'm going to give you some more examples of that, and then talk about the various ways. We haven't said much about how repressors work. Okay, so let's talk about repressors first, because that's what's up. How do they work? Okay. Well, one of the ways they can do is if the enhancer sequence overlaps with the silencer sequence. And what that means is the footprint to which the activator and the repressor bind is going to compete. They're going to compete for the same bind. They're steric and hindrance. They can't both sit there. One is going to win, and the other one is not going to win. Okay, so they're competing for the same binding site. So the repressor can outcompete the activator depending on the strength of binding and what the concentrations of activator versus repressor. If there's lots of repressor being synthesized, that's going to turn the gene off because it will win the battle. Right? And if this is a fairly weak activator, there can be equal concentrations of activator and repressor, and the repressor will still win because it's got a stronger attraction for that particular element. It all depends about the strength of attachment and the number of transcription factors present. Who wins this battle? Okay, so competition, competitive inhibition is the way you should think of that. Okay, a second way is they bind to unique control elements, and that's what the generic term is for pieces of DNA. Okay, and one's here and one's here, but this activator is supposed to interact not with the repressor, it's supposed to interact with what? With the pre-initiation complex. But if this blocks, if this repressor, and what domain is this? What do you call this domain of both of these guys? It's the activational, transactivational domain. That's the best name for it. Even if it's repressive, you don't call it the transrepressional domain. It's transactivational domain is interacting with the transactivational domain of this repressor, and therefore it's competing with the binding site, so this cannot talk to the initiation complex. It prevents interaction. Okay? So that's a pretty clear way to do it. Okay? And the third way is, what if the repressor activational domain blocks the site where this is supposed to bind? So it can actually bind to one of these, this is a general transcription factor, and if it blocks the binding site for this activator, it's no-go. Okay? So those are three basic ways that you can block transcription. And here's a more subtle way. Okay, let's say this is it's red, it's a repressor, let's say, and it binds to a silencer. And when it binds there, it recruits a type of remodeling complex. Now, let me get this out of the way. Sorry. A type of remodeling complex which is going to tighten the compaction of DNA and turn it off. So it's going to turn it into a 30 nanometer fiber that is too packed in order to cause transcription. So it starts to cause heterochromatin formation. Okay? Or it could recruit what would this protein be? This is a repressor, so it's not touching DNA, so it is a co-repressor. Good. Okay? And a good example of a co-activator would have put these guys on, which would be a hat. Histone, acetyltransferase. Okay? So if you deacetylate all these lysines, then you can do what to them? You can methylate them, and that usually turns them off. Okay? Not always. Okay. And then it can also directly recruit another co-repressor, which would be histone methyltransferase. That doesn't always work, but if it's this compact, it's probably going to pack it even tighter. Okay? So you can use epigenetic repression as well as those transcriptional repression mechanisms. So epigenetic control and transcriptional control are joined together. They work together. All right, so regulatory proteins bind cooperatively. We talked. Okay, so here we go. So this is a really simple slide, and there's a couple of really important concepts to take away from it. We look here at the construction here. These are two different genes, and the reason they get uniquely turned on in the correct situation is that this combination is not the same as that combination. Do they share components? Yes, this is the same as that. Okay, so they share the same components, but why does this gene turn on differently than this one? It's because these two guys are unique. This co-activator and this activator are very different from the surface that is presented by these three guys. Okay? And this is a way oversimplification simplification because it's not 3 proteins, it's dozens. Okay. So they make a unique dog pile that somehow both of them stimulate the genes, but only in the circumstances where all 3 of these guys are present, even if they share some of the same transcription factors. So it's a unique combination of transcription factors. Same thing goes with repression. You can have 2 different groups, and the reason 2 different genes this gene is turned off and this gene is turned on. Why? They both have this little red guy here, but it's the unique combination. And this one is normally found as an activator. But in this context, this unique combination recruits a co-repressor. This combination creates a shape. It's all about the protein shape. That recruits a co-activator, and that's going to turn on. This one's going to turn off. So going back to our steroid receptor example, sometimes the glucocorticoid receptor interacts and recruits an activator, like a hat. And it acetylates the histones, and they uncoil, and it opens up, and the gene transcription is stimulated. But sometimes the glucocorticoid receptor, it loves to turn off the immune system and shut down inflammation. So it inhibits a large variety of genes. How does it do that? Well, it's sitting there, but it's going to recruit a co-repressor and not a co-activator. So maybe in the immune cell that it's targeting, like a lymphocyte, maybe what it does is it recruits an HDAC instead of a CAP. It's a really straightforward mechanism. This seems really simple, and it's a core concept to understand. Okay, how are we doing? Everybody? Okay, so let's look at a really straightforward example. Thank goodness Drosophila is very simple, but this type of switch is very similar to human genetic switches. So we're going to look at this in some detail. Okay, so you need a little bit of biology. So this is the egg a couple of hours after fertilization. And what happens is, it's kind of weird, it makes a bunch of nuclei up here, and this is all one cell. So it's just the nuclei that have been divided. Okay, and each of these nuclei are going to synthesize a unique concentration of a transcription factor. Okay, bicoid and hunchback are both. They control the anterior part of what develops out of that egg. So it's going to control the head and antenna and part of the thorax, the front legs. Okay, and so what's going to happen is their concentration of bicoid and hunchback is higher at the front part of the animal. Okay, now these 2 guys are both repressors. Giant and Krupel are repressors, and they have a very unique distribution compared to these guys. So how does all of this work together to specify where the body parts are going to go? Well, all of these 4 guys are used to control the expression of 7 different stripes. And each one of these stripes is a stripe of protein called Eve. That comes from the word even skip is the gene. Even skip, not surprisingly, is a homeotic gene. And you know what a homeotic gene is, right? It's got that 180 base pair that codes for 3 different alpha helices. It's got a helix turn helix. It's going to insert into the DNA and tell this protein where to go. It's going to tell where the head should be, where the antenna should sprout. So you have these really orderly stripes of Eve that tell the body plan where to go in this embryo. So what's cool is, so this is what the actual structure looks like. You have a group of control elements that controls a single gene is often called a module. Okay? So it takes all of the control elements in this module to set up where the stripe goes. So this one we know sets up where stripe 3 is supposed to be laid down. This one controls stripe 2. This one controls the position and concentration of stripe 7. But look, they're all acting on the same gene. It's just depending on the unique combination of transcription factors will activate this module and not that one or this one. So let's take a really clear example. We're going to take one of these modules and we're going to put it on a piece of DNA that we've constructed. Okay? We're going to use the same type of promoter that would be used in Drosophila, but we're going to get rid of the E gene. We don't even care about the E gene. What we're going to do is replace it with something that we can measure easily in the lab, which is the LAC-Z gene. Okay? That I told you, if you put the substrate in there, it turns a blue color, the gene turns on. And when we just get rid of all the other modules, what's the expression going to look like? Well, this is the way, this is actually immunocytochemical stain for Eve, and then there's stripe number 2. This is the one that we constructed, and it's blue because that's the color that LAC-Z produces when it's turned on. And it doesn't exactly line up, but there's one. It's this stripe right here is this thing right here. And you notice there's no other stripes because there's no other modules. To put a blue stripe there, or blue one here, blue there, you could do that. But we're going to focus right on that. Why is that stripe right there? That's our question. Well, we know the answer from your concept. It's a unique combination of those transcription factors. Okay, so this is what the module structure looks like. So the first thing, let's do the key. All the repressors are on top of the gene. This is just how the cartoon is organized. This is not real. Okay. So all the repressors, so Cruple and Giant are up here, and then Bicoid, which looks, Bicoid looks like a banana, and this looks like a really sad T. Okay, and so these guys are underneath, right? So just to show the display. Where are the control elements? Okay, well, let's look at Cruple. Cruple has 1, 2, 3 control elements. But notice that control element overlaps with 1, 2 of Bicoid. Okay? So you can already get a guess that those two, that repressor is going to compete with Bicoid at those 2 sites, 2 out of the 3 sites. Okay. Now let's look at Giant. Giant has 1, 2, 3. And Giant interacts or competes with Bicoid here, and it competes with Hunchback there. So there's all this repression is going to be mediated by competition. So the concentration is going to be really important of the transcription factor. So that's the first hint we got. Okay. But there are unique places. This one, Bicoid can bind without any kind of competition. Hunchback has only got one place to bind, and there's going to be a really important consequence of that. So let's keep that in mind. So this is probably going to be the likely main mechanism of repression. This is why the stripe is not going to be in position number 1 or position 3, because this repressor is going to keep Bicoid or Hunchback from doing their job, which is to turn the Eve gene on. Okay, how are we doing? Straightforward? Okay. Now, you need another piece of information. The reason that position is laid down at that stripe number 2 is the combination of the 4 concentration profiles. Okay, so let's look at them. Okay, it makes sense that Hunchback and Bicoid would be present, right? Because they're activators, and that's what would turn the gene on. Okay, and notice, though, they're both present, but which one's higher? Hunchback, right? You have to have high concentrations of Hunchback, and you can get by with just low concentrations of Bicoid. Okay? Now, look at what the rule is for Giant and Cruple. They have to be gone. And we know the reason for that, right? We can't have them competing for those control elements. They get in the way. So the reason that stripe 2 is laid down there is because of the absence of Giant and Cruple and the presence and the correct concentration of Bicoid and Hunchback. So just 4 different proteins, and this is a very simple case, is going to control where stripe 2 is laid down on the embryo. Now, the other thing, so we haven't answered one question, and your book doesn't answer, but I'm going to answer it, because why is Hunchback have to be so high in concentration and Bicoid can get by with being low? Is it because they're competing for the same sites? You have to look back at the key. No, this is Hunchback. It's only got one site, and it's not competing with Bicoid, so that's not why it has to be high. What's another difference between the control elements for Bicoid and for Hunchback?
Hunchback occurs at less often.
Exactly. So the concentration is decoded by the number of control elements. If you have only one landing site, you better put a lot of hunchback in there so it finds that one spot. But Bikoid can be at low concentration because they can bind there, it can bind, so this is gone, all these guys are gone, so it can bind there, 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 there. Low concentration is appropriate if there's lots of control elements to which it can bind. That's how concentration is decoded. Not a word in your book about that, but that's key to understand. Okay. Okay, any questions about what? Yes?
Does Bikoid need to be positioned on each of those elements, or can it be like on two and it would work?
It's probably the case that it works more strongly if all five are occupied, and it has an easy time of, or it can do it, but I think it will go, it'll be okay if it just finds a couple of them. That's why you can get by at low concentration. So the answer to your question is it doesn't need to occupy all five. Okay, good. So that's a really basic example of combinatorial control. Repressors interacting with activators, or they have to be absent in order for there to be activation. Okay, so good example. That's the whole point. Oh yeah, why did you need both Bikoid and Hunchback? Because of this phenomenon. If you got them both, transcription will really synergistically increase as opposed to the individual increase by themselves. So that's why you need two. Good principle. Okay? So the sort of take-home lesson is the probability. This is just a probability. It depends on who's present and when they're present and how strongly they bind to this transcription site. Do you have an enhanceome somewhere located here that bends the DNA to bring these control elements close enough to talk to the pre-initiation complex? You have to have enhanceomes that bring these distant control elements close together. And it's not the control element that does the interaction, it's the proteins that bind to the control element. You can bring all kinds of DNA all day long, it won't do anything if you don't have a protein stuck on it. Okay, and so you shouldn't take my word for it that mammalian switches work the same way. So let's look at a really well understood one, which is the beta globin gene. And what is beta globin? It's what? Subunit of hemoglobin. So hemoglobin has 4 subunits, so it can only be described by quaternary structure. It's a heterotetramer. It has 2 betas and 2 alphas. We're going to only focus on one subunit. So this is the gene structure for the beta globin gene. So it has a typical structure. It has, so here's a gene control region that's really close to the start site. You can tell where the start site is because it's where the first exon begins. Remember, you have to begin and end with an exon. Okay. Then you have, that's kind of an unusual intron. Why? It's small. It should be 3 or 4, 5 times as big. This is a typical intron. Okay. And this is the last exon. And of course, this is where you're going to put the 5' cap onto the transcript. This is the DNA, of course. And then this is where the poly A site is the site not only of poly A addition, but it's also the cleavage site. RNA pol 2 is going to just keep running. The termination site is somewhere out here about 1 to 2,000 base pairs. It's sort of weakly indicated. So pol 2 always goes, all the polymerases always go past where the cleavage point is. Okay? And that's because you need to have proteins to identify, bind to the transcript and go, oh, this is where we're going to cleave it, right here. So the termination site is different from the poly A cleavage site and addition site. Okay, so that's just the basic structure. Let's look at this is like the Eve stripe control module. You pull it out, and you notice there's a bunch of transcription factors. Some of them are, this is a very common transcription factor that binds to a lot of different genes. So here's one of our common ones. But it's the unique combination of this guy and the GATA1, particularly GATA1, is very specific to erythroid cell lines that produce red blood cells. And so GATA is one of the unique proteins that is there. But it's in combination with some of these proteins that are in several other different genes that help turn those on. Notice that the control regions can be upstream, they can be downstream, they can be way past the termination site. It doesn't matter, okay? Because this DNA can loop back and interact over here. They can be located within introns or sometimes exons, but typically introns. Or they can be way off the screen here, and they can be in a different chromosome. So that's what we're learning. These control elements are bizarre in their orientation. Okay? That's the only message from this. Okay? And so this shows, again, this is sort of like when do you get, why did you get strike 2 at that location? When do you get, so the parallel question is, when do you get the beta globin gene turned on? Well, it's expressed really lowly in utero. So this is in the embryo. And so you have unique types of globin genes expressed. And why would you want a different type of globin gene in a developing embryo compared to you and I as adults? Is the environment different? Very low oxygen, right? All the oxygen has come through the mom, go through the placenta, and then, so these guys have really high affinity for oxygen. And so this one has some other properties that make it unique and special for use in adults. Okay, so we want to turn this guy on. This is what here's the cluster of genes. So now these are not control regions. These are the cluster of globin genes. Okay? And the one we want to turn on is right there. And so remember, if we flip back to the other slide, you see that it has a control element here. It's got a control module downstream. And then it's got this guy. This is called a locus control region. And this is very typical of mammalian eukaryotic genes. This thing is located like 100,000 base pairs upstream from all of these genes. So this thing is going to loop out and control and help turn on each one of these genes at the appropriate time. And the locus control has 3 properties. Number 1, it's usually located a long way away because it's got to be able to arch up and touch this one, arch up and touch this one, that one, that one, and that one. So it's usually located a long way away. Second thing is it uses 2 mechanisms to activate transcription. So it's going to loop up here. Let's look at the beta gene. It's going to loop here and attract the appropriate transcription factors, mediator, pre-initiation complex components to sit down on the promoter. So it attracts positively all those components that you need. The second thing it does is it prevents heterochromatin from invading this region. Okay, so it has a barrier function. So if, say, this gene starts to become heterochromatized, and you remember that's sort of a amplification, sort of a moving process, you need a barrier to keep that from turning these genes off. Unless you want to turn those off. When would you want to turn these guys off? Let's say you're in a stem cell that's going to give rise to the erythroid line. These genes are there, but you need to be undergoing cell division, cell division, cell division. So that means you've got to package the chromosomes into mitotic form. So that's when you would heterochromatize this whole region and pack it up and ship it off. Right? FedEx it. Okay. Okay, and then there's one other type of sequence that we'll talk about before we take a break here. And that is, so we talked about barrier sequences, and the locus control region has a barrier sequence function, so it can play this role and prevent this heterochromin from invading gene B or gene A. But there is also a different type of control element. It's called an insulator element. And it's located near a place like an enhancer or a locus control region, And so you know this enhancer loops out to interact with this gene B. That's the appropriate interaction. So there's no insulation between the contact of the proteins assembling here and the proteins that assemble there. They're free to do that. Okay? And we're keeping the heterochromatin at bay using a barrier. And then what happens, though, is this enhancer could have proteins that loop out and then accidentally flop over here and talk to gene A. That would be inappropriate. So the insulator element competes for binding with these enhancer elements. It insulates the enhancement towards the correct gene and keeps it off an illicit gene that does not need to be turned on. So you see the difference between the two? This is to prevent heterochromatin from encroaching. This is to prevent the wrong gene from being turned on. So insulator versus barrier is a distinction. Okay. Questions? All right, let's take a break here.
I have a question.
Yeah.
But when you said that the start side is indicated by the exons and on
The present of the first exon.
Sorry?
The first base pair of the first exon,
Yes.
that's where the start site is. It includes the +1.
Okay. Yes.
Yeah.
But I'm getting confused on for some reason. I thought this is going to be after the DNA replication, after the introns have already been cut off, correct?
At what point? I mean, you can look at a gene and you can identify, oh, that's going to be the first base pair that's going to code for the transcript. Because it's turning it, transcription turns it from a DNA sequence into an RNA sequence, right? A complementary RNA sequence. So you're reading DNA, you're making RNA, but you can look at the gene and go, that's going to be the first complement. So if it's usually an A, so that means usually the first base of the transcript is a T, right? Or actually, it can't be a T, it has to be a U because it's RNA. So you can say that is going to be the first base pair and it's going to be an exon 1. It's going to be the start of exon 1. So the gene structure that I was showing, that's the DNA, but you can tell where it starts, right? Because let's just go back and find it. So you look up here, and this is sort of your way. It's 400 base pairs. This thing is your way out. So you look at this, you go, where's the start site for this gene? You find the first exon to the left, and you go, that base pair right there is where it's specifying the first nucleotide to make the RNA. It's always the first nucleotide. That is. So where's the poly addition site? This is the last exon, so the white thing. It's the right-hand side of that exon is where poly A is going to be. But the other thing you know is that the 5' translated region is going to be at least part of this if it's not perfect. This is going to be the last exon is going to be at least include a fraction of the third prime. It's the other. And those are actually controlled.
Okay.
Sure.
I'm confused about the bicoids and the hunchbacks.
Okay.
So we have
They're both activators.
They're both activators. So
same class of
is it pretty much one is a stronger activator than the other? That's
One
why
can be as stronger, but their control elements differ in their presence.
Okay.
The ones for hunchback are rare.
Okay.
The ones for bicoid are all over the place.
So the makeup, because it's so rare, is kind of complicated, but it's stronger because it's so rare.
Well, no, it's not stronger. It just means that it requires a higher concentration of the protein, or else it won't find it. See, this is a noisy place, And even if you're in a transcription neighborhood,
Right.
there's all these genes that are. I have no idea how that works.
Okay.
But it's complicated, and it has to find its own control element. And if it's rare, you better have buckets.
Okay. So would it like most of the time win like that competition with like
The repressors?
Yeah.
No, they need to be gone. They almost always win.
the hunchbacks?
The
repressors.
Krupel and Giant.
Okay.
If they're there, the gene doesn't turn on. So they must be really strong or have high concentration. But if they're absent, then you don't even worry about it. And so in this particular scenario, there could be a scenario where you're talking about where they're both present, and there's competition, and the activators win because of the concentration and strength of binding. That could happen, but not in Stripe 2.
Okay, cool.
Stripe 2 is that's why I chose it, is because the book chose it. It's really straightforward why the stripes laid down there.
Okay.
No repressors, adequate concentrations of both activators. You get a synergy that turns on lots of E.
And when you use LAC-Z and it turned blue, was that significant?
Well, it is significant because why doesn't that affect the transcription if you put a different gene there? No, it doesn't. It has no effect on the transcription because apparently the Eve gene has no control elements located in the DNA. If it did, it would slow down when you put that LAC-Z there because that's it's beta-galactosidase, which is an enzyme that takes a substrate and turns it into a blue product.
Okay.
We do that so we can see it because we're too lazy to do the immunocytochemistry. The immunocytochemistry is expensive and it's time consuming. A blue color you can do up in our lab, up in the cell lab. Yeah. Yeah. Were you Daniel?
Yes. Were you start presenting Wednesday?
Oh, no. I just want, so the due date for everybody, and I'm not going to break that, but it's easier for you if you go ahead and find your free papers and figure out which one you're going to do. Because I'm trying to give you guys more time.
Alright, I just confused. I was like,
No way. I haven't changed the date. I thought about delaying the week. The schedule's just too tight.
that's how I feel.
That way, see, next week, this week, you can forget about the test for a day or two and just go to the library, go online. You can find it.
Yeah, I was looking yesterday and taking them home. So
Yeah. In fact, if you can just save yourself some time and get the PDFs, that does two things. It allows you to have the paper. So when I say, oh, that's what I want, you can just send it back to me in a matter of seconds. And second of all, you can read the abstract or look at the paper to make sure it's a primary literature, not a review on that. That's the other thing. All right. Good deal. Yeah.
Okay, I just have a question about looking back to the lectures, talking about kinetics. And, okay, so you have one catalytic
Yeah.
there can be properties used with regulatory circuits, so that you do the same.
It gives you, that's a good question. It would create a signal, because you can't get a signal. The book shows you there's a sigmoidal chain. It depends on how you plot it. It wouldn't look the same way we did. The book plot, you notice that it changed the scale. It's no longer initial rate versus concentration. It's repressor concentration. I just didn't want to say so I wouldn't ask you that question. It's really easy to get both feet deep up to your, up to your hips and stuff. I'm trying to say, if you get the basic concepts, you can figure that out.
Okay.
But my guess is that it would affect the curve and displace it from what you would expect, which is completely. So if you had no regulatory subunits at all, or if they're present, but there's no regulators present, you've only got substrate, no signal. That's a question I can ask you. Say, you've got 6 regulatory subunits. You've got monomeric catalytic subunits. No, what do they call it? Allosteric inhibitor. No, say CTE. Well, the curve, what it would be on the is you have it can't react if it has no binding target. So the regulatory subunit shape cannot change.
What if it just what if there's not proximity between the regulatory subunits because there's only one regulatory subunit?
That would not be significant. probably would, what it would do,
It would change the shape of
the position of hyperbolic protein would still be hyperbolic.
Okay.
It would probably look like competitive inhibition.
Thank you.
Okay, let's get started again. And I'm sorry I'm not able to show you the outline. I just forgot my laptop, so it's been a rough week. Okay, so let's figure out where we are, because this can get really out of hand very quickly. We've been talking mostly about this, but we've integrated the epigenetic controls with transcriptional control, and then we're going to move very quickly past this because you're supposed to already know this. I know you don't, but we'll talk about it very quickly. Then there's transport control into and out of the nucleus, and so there's all these other things that we need to talk about. So one other thing we need to talk about is how do you control the concentrations and activity of transcription factors? I mean, they have to be made, correct? So that occurs out here in the cytoplasm. Now remember, when I'm talking about proteins, think about where they work and where they're made. That's two different places, two different compartments. One of the control steps is this transport through the nuclear envelope. So all transcription factors are made in the cytoplasm out here. They're not made in the nucleus, they're made in the cytoplasm. So that means getting in through this little portal is a point of control, gene control. And let's look at how, that's just one way of controlling it. So let's think about our example of the steroid receptor, and we'll talk about the glucocorticoid receptor that binds cortisol, okay? So one way of controlling the amount of glucocorticoid receptor is to control the synthesis of the protein itself, because the glucocorticoid receptor is a protein, so you can induce or turn off the gene to control the amount of transcription factor. This is a very basic way. It has to be there or it can't be involved in the control. Second of all, it has to go into the nucleus, okay? And that's kind of an interesting story. That's driven by the binding of the hormone, so it's cortisol to the glucocorticoid receptor, it's estrogen to the estrogen receptor, it's testosterone to the androgen receptor. So it's the ligand has to be there to activate it. So ligand activation is classic for steroid receptors. So this is really obvious. Covalent modification. The covalent modification is typically phosphorylation. So the glucocorticoid receptor gets phosphorylated to help it get rid of some inhibitory proteins and go through the nuclear pore. So phosphorylation is a very important switch to activate transcription factors. Very important. Lots of kinases turn on transcription factors that are already sitting on the control element. They're just silent. They can't work until they get phosphorylated. So phosphorylation, a covalent modification, is very important in the glucocorticoid receptor. Addition of a second subunit. Glucocorticoid receptor, as you saw, requires two different subunits. It is a homodimer. Each subunit has two zinc fingers, and they do different things, as you remember. But you have to have two to sit on the DNA to turn on the gene. And you have to have both of them be correctly phosphorylated in the right spots to get in through the nuclear pore and sit on and find the, what a response element? Glucocorticoid response element, okay? So subunit addition is very important. And if you have the wrong subunit, you can have inhibitory subunits. Most of the time, when you don't want the glucocorticoid receptor activated, let's say you don't want your blood glucose to go up, you want it to go down. Well, what happens is a group of proteins called heat shock proteins, these are chaperones, okay? Heat shock protein, particularly heat shock protein 70 and 90, bind to the glucocorticoid receptor and they mask it. The only way to get rid of that mask is, well, there's two ways. Number one, the hormone binds, so it sort of shakes itself off, and the heat shock proteins, we call them HSPs, heat shock proteins. HSPs dissociate, and so that allows them to be phosphorylated, and it allows them to dimerize and go through the nuclear pore. Okay. Usually they dimerize before the nuclear pore. Okay, so masking, that's a great, the glucocorticoid is a great way to talk about that. The masking protein is a heat shock protein. Okay. Stimulation of nuclear entry, two things have to happen. You have to have no heat shock, well, let's not go that far. You have to have the inhibitory heat shock proteins gone, and you have to be dimerized to go through the nuclear pore. Okay. So different transcription factors get to go through the nuclear pore depending on what that particular transcription factor is. For glucocorticoids, they need to be dimerized, and they need to get rid of most of the inhibitory heat shock proteins. Okay. And then there are cases where, this one we'll sort of ignore, but we actually do know of cases where glucocorticoid receptor is actually in an isoform that's attached to the membrane, and the way you activate it is to cut it. You actually proteolytically cleave it, and then it can go into the nucleus. So, really complicated. It's not just about synthesis, it's about location, dimerization, masking, phosphorylation, ligand activation, transport through the nuclear pore. This gets really noisy, okay? But it requires that exquisite level of control to properly turn a cell on or off. Because if you turn the cell on or off in the wrong context, it's damaging. If you suppress the immune system so badly that the infection takes over, you just killed yourself. Okay? So that it's really important about the control issue. Questions about that? Okay. All right, so we come to the last control issue. We're not going to go through all of them on the Quiz 1 material. So this is the last segment that's going to be on Quiz 1, and that is alternative splicing. So where do we put that? Let's see if I have a category. Let's go back here. That would be right here. That's RNA processing control. And splicing begins co-transcriptionally. As soon as the little bugger shows its 5' head coming off of RNA pole, it starts to be, the spliceosome grabs it, it starts to cut out the introns and splice together the exons. The tricky part is it doesn't always work the same way in different cell types. In one cell type, it will take out a different set of introns. In a different cell type, it will splice together a different set of exons. So that's called alternative splicing. And I hate the example that your book gave you, so I'm going to talk about my own example, which strangely enough is a hormone. But let's talk about the basic part here. So you have primary transcript, you get rid of the intron, you splice the two ends together. These are actually spliced together by a ribozyme, not by an enzyme. The proteins are there to hold the RNA ribozyme in the correct position to cut and splice the RNA. Okay? So the way you get alternative splicing is you now need to think of that new transcript is it's a gene product, okay? But it's got its own response elements. It's got its own control elements, and it's got activators and repressors that bind to it. So repressors and activators and control elements is not a DNA thing. It's a nucleotide thing. So we've just made this, and what we have here, this is an intron, but this is a splice repressor. So it's actually binding to a splice silencer at that sequence. And it says, I want you just to skip this intron. So we're going to synthesize, that exon is going to be synthesized. This one's going to be this is going to be synthesized, but instead of cutting it out, we're going to treat it as if it's part of this exon. So you can actually leave exons in there, and it will create a new messenger RNA from the same gene. Okay? The other thing is also possible. You have no splicing in one cell type, but in a different cell type, you can use the same gene, and an activator binds to a splice enhancer. So you have control elements in RNA, and we'll beat that concept to death. So don't forget, there's control elements in RNA just as well as in DNA. It shouldn't be too weird. Okay, so it's hard to sort of put this in a context without a real example, and so I'm going to talk about the calcitonin gene, because I hate the one that your book uses. So we'll flash that off. Okay, so let's just draw, this is the calcitonin gene. Is that in focus? Yes. Okay. And calcitonin, it's famous for being produced by the thyroid, but it's not a thyroid hormone. It's come from cells that are attached to the thyroid follicle, and what calcitonin does is lower your blood calcium level. Now, why would you want your blood calcium levels lowered? Well, the body is very careful about regulating calcium levels very tightly. Because if it goes too high, you go into some sort of tetany, and if it goes too low, you have the opposite problem. Your muscles don't contract, you can't breathe. So calcium is highly regulated. Third, but the other cool thing about calcitonin is what it does is the way it lowers blood calcium is to create more bone. So it is a very good osteoporosis treatment. And in fact, what they give in the pharmacy is not human calcitonin, it's salmon calcitonin. That sequence is highly conserved, but because the fish calcitonin is a little different, it lasts a lot longer in your blood, and so it can do its job, which is to strengthen bone. Okay, so that's the physiology and endocrinology, and this is the structure. It's got, let's just put a bunch of boxes here. These are going to either be introns or exons. Okay, let's see how many boxes we've got. All right, so this is exon 1, 2, 3, 4, 5, and I think there's 6. So I need one more. Okay, so E6. And everything in between these are introns. Okay, I don't know why I did that. All right, so what happens is in the thyroid, you're going to make calcitonin. Okay, and to do that, what you do is it takes exon 1, 2, 3, and 4, and then there is, now this is where it gets complicated. Not only is there alternative splicing, but there's an alternative poly A tail site. And it's going to choose this one, poly A tail site 1. And it's going to cut it here, so it takes this transcript and it's going to cut out that. And so the protein, or the RNA, messenger RNA, looks like this: E1, E2, E3, E4. So it's missing all the introns, but it didn't go past this first alternative polyadenylation site, so it's only got 4 introns. And that makes, and then when this is turned into protein is calcium-lowering calcitonin. All right. Now, what happens is that's the thyroid. So in a different cell type, you can have a unique set of splice enhancers and repressors. These are proteins, and they're made. It's the same gene, but they're going to do a couple of different things. Number one, so we're going to make a different protein, and it's whoever named it was a really boring person. Calcitonin gene-related protein, peptide. Calcitonin gene-related peptide, CGRP. Sorry, it's ugly. Okay, and this is found in nerves and has an entirely different function because it's got an entirely different protein structure. And so what it does is it decreases gut motility, so it's a gut motility regulating hormone. It decreases acid and pancreatic juice secretion. It doesn't do anything that calcitonin does. And it increases blood flow, so it's a vasodilator, lowers blood pressure. That's why pharmacology people are interested in it. So there's all this, you don't need to know those functions, it's just that they're different from calcitonin. Different physiology because it's a different function. Same gene though. Okay? So how do you get calcitonin gene-related peptide? What you do is you use an alternative poly-A site number 2. And so it ignores this and it cuts it there. Okay. So it takes site 2, so now you've got 6 different exons you could splice together. But that's not what it does. What it does is it does everything else. It goes E1, okay, we'll take that, and I'll take a little smidgen of E2, and I'll throw in a little salting of E3. But you know what? I'm going to cut that out, but I'm just going to pretend that this exon and E4 is an exon as well, and this is an exon. I'm sorry, intron. So it takes this intron, that exon, and this intron, and treats it as one big intron. So what has it got here? It's got this is a splice repressor that sits right on top of E4 and keeps it from being, yeah, that's right. And so this whole thing is cut out. So it should cut all these 3 pieces together and stick it in there. So E4 is missing. And so you have E5 and E6. So that's the structure of calcitonin gene-related peptide. It's longer, but it's missing one of the characters. And so that makes the CGRP peptide. Where? In nerves. Any questions about that? So it's a combination of alternative polydentylation and alternative splicing to create 2 different messenger RNA. So now, if this is true, and are there any questions about this? Yeah.
So you said it was a splice repressor that caused that to get out of the splice.
It's a splice repressor because it just leaves it as part of the intron instead of cutting at the borders. So that's why it's confusing. But if you remember that it's left in there and then you take out that whole gigantic chunk, you get rid of exon 4, that's the key point. Not to figure out whether there's introns, exons, or enhancers or that sort of thing. I'm not going to ask that. Okay. Where's my look? I'm looking for my control element. Okay, so let's generalize a couple of things. The first generalization is we have to change the definition of a gene, and we've known this for some time. Actually, most genes are alternatively spliced. So an updated definition of a gene is a sequence of DNA transcribed as a single unit that encodes a set of closely related polypeptides or RNAs. That's the only difference. Do you have? So what do you call? This is a gene. It's a type of gene. What is the transcription unit called? It's called a complex transcription unit. If you get more than one RNA type of RNA from the same gene, and it's complex because in this context you get this transcript. In this context you get that one. It depends on the tissue context. Okay. What do you call one that you just get one RNA all the time? That's a simple transcription. Makes sense, right? Okay. And so let's get the full picture now that you've seen examples of where you use alternative splicing. So you can get complex transcription by several different mechanisms. You can choose. These are the 2, B and C, would be the alternatives the calcitonin gene uses to get 2 different proteins, peptides actually. Okay. But you could also have alternative promoters. What if you had a promoter that started upstream from this promoter? So the one that's closest to the start site is going to produce a smaller piece of RNA because it's starting closer to the start site. So the alternative promoter could be way upstream, and the start site's very different. And you might have to cut out the first promoter, but you can have different promoter sites. This tissue starts here, and this one starts here. So that produces an RNA of different length. Okay? And the alternative, the exact opposite, is also true. If you select a different termination site, you can make RNA shorter or longer, depending on where you terminate that transcript. Okay? And it's not where you put the poly A tail, it's where the termination site for the pole 2 is. So under one context, if you terminated, say, 500 base pairs past the poly A site, what's going to happen is it will use that poly A site. But if the transcript is 2,000 base pairs distant, it might pick a different poly A site, and therefore you would have to actually have both of these two things working together to create two different RNAs. Okay, so it's complicated. Okay. Questions about that? We go on. And so that's the end of quiz 1 material. Because we just sort of completed we're now about to shoot out into the cytoplasm, and a whole different set of problems arise. Okay. That's the end of that. We do have a few minutes, and since I got started late, I really do need to use them. So this is lecture 7, which will not be on. So this is a real nice demarcation. It's lecture 7 is on the new material, lecture 6 and previous to that is on quiz 1. Okay, so now we're going to pop out here into the cytoplasm and talk about, we've talked a little bit about this control point, which is getting through the nuclear pore, but until we start talking about trafficking later on, protein trafficking, we're not going to explicitly do that other than that example I gave you with the glucocorticoid receptor. Remember, you have to get rid of the heat shock proteins and dimerize, and then you can go through the nuclear pore. But that's the only control I'm going to mention. Okay, so the first thing is messenger RNA itself can be transported to different locations, and that will change the location of where the protein is made. Why is that important? It seems like it could just diffuse all over. That's not the case. So some messenger RNAs are localized to specific regions of the cytoplasm, and that's particularly true in our old friend Drosophila. This is one big multinucleated cell. These are the nuclei, and this is the cytoplasm. The reason that bicoid is where it is because when these nuclei make the bicoid messenger RNA, it's carted up to the front of the animal. How does it get there? So it spits out into this cytoplasmic soup, and then there's a protein that binds to the 3' untranslated region. There is a response element that protein binds, and that protein will link it to microtubule motors. So it's going to bind to a microtubule, and the microtubule, as you know, is a transport mechanism. It's a highway that you can carry vesicles and mitochondria and all kinds of trafficking of particles. In this case, you're actually trafficking a messenger RNA along the microtubule. So remember the movie, The First Day, there's that Keynesian guy walking along. What was he carrying? It's a big vesicle, right? In this case, he's carrying a messenger RNA. Where is he touching it? The 3' untranslated region. That's where most of the messenger RNA transport control is invested. Now that makes a lot of sense, right? Because you can, if you put the information for moving the transcript in the 3' untranslated region, it's not changed into protein. So you can have the same instruction as move me to the front of the animal, in several different genes, but it never gets coded into protein, so you don't have similar proteins. You have a unique signal that says, move me to the front, but it's not part of the protein that's going to come from that messenger RNA. It's just for transport. So let's put it in a part that doesn't get transported, doesn't get translated into protein. Put it in the 3' untranslated, or you could put it in the 5'. It just turns out that typically transport recognition by the cytoskeleton is in the 3' untranslated region. Okay. It's a motor protein. Okay, so what's going to happen is it's going to ferry it to this part of the animal, and then once it gets there, that messenger RNA is held there, because otherwise it would diffuse back slowly. But this gradient needs to be set in stone, or at least in fly, and stay there. Or else you're going to have a screwed up, the antennas coming out of its butt. It's just, this is just wrong. It has to, development is highly evolved because it creates a monstrosity if it goes wrong. Okay, so what holds it there? It binds to the actin cytoskeleton. So the cytoskeleton is controlling the transport, microtubules, and actin is binding it and holding it there. Okay, and so you can create lots of different patterns using that type of system. So here's the generalized version of it. Okay, so you have this nuclei is producing these transcripts, and this is what happens with this is actually bicoid. Bicoid hitches a ride on the microtubules. It gets up to the anterior pole of the cell, and then it jumps off the microtubule and binds to an actin protein. So it's held, localized, so it takes both of these mechanisms to work. Now, you could have a system if you weren't under the crush of time. Remember, this fly embryo is going to create a fly really fast because they're going to live for 24 hours and they're going to die. So things happen very quickly. And so this is a very fast way to produce a larva. Yes?
Does it also, like when it binds to actin, does it bind every 3' as well?
I think so, but I don't know the answer. I'll find out. I don't know who to ask. Let's pretend for the time being that it does. Okay. You could just randomly produce this stuff and it could diffuse, and if you just had the actin binding system, it would still localize to the anterior pole, but it would take longer. That would be the penalty for that mechanism. So this uses a combination of transport along microtubules and retention at a particular place. And then what you could do is you have them localized here, and everybody that's not localized gets destroyed. So selective degradation. This actin cytoskeleton is protecting it from degradation. And so this probably, there's a combination of all three in place in almost every messenger RNA that's transferred. Okay. All right, so we'll do that next time. I think that's enough for today. He was here, he forgot to sign in. He didn't bring a clicker? Okay. So we can do it.